Hello and welcome back, future scientist. I'm Mr. K with Mississippi Public Broadcasting, and I have another lesson ready today talking about the cell cycle. So today I wanted to talk about mitosis, and we're going to kind of get into the details of this, kind of get into the really specific stuff. Last time we did talk about mitosis, but we were just kind of looking at some pictures and just kind of introducing it. So today we're going to get into those really specific details and see what's going on in the cell in these phases in a little bit of detail. So just as a reminder, what is mitosis? Well, mitosis, it is the division of the nucleus. That's really what I want us to remember here, okay? Mitosis is the division of the nucleus, and our goal here is that we want to end with two identical nuclei, and what that means is we're specifically concerned with the DNA inside of the nucleus of these two cells. Remember that for a cell to do its job, it has to have DNA, and that DNA copy has to be exact as what we started with. So we're starting with one cell, we're going to duplicate all the DNA in that cell, we're going to divide it into two cells, and the goal is by the end, those two cells need to be identical to what we started with, okay? And mitosis is how we make sure that that happens. So just as a quick reminder, mitosis, there's a lot of stuff going on in mitosis. And so we break it up into 4 different sections, and this makes it a little bit easier for us to study what's going on. So the 4 stages of mitosis are prophase, metaphase, anaphase, and telophase. Okay, and a really convenient little acronym to remember so that you can put these in the correct order is PMAT. If you can remember PMAT, that's going to help you put these 4 in the correct order so that you're not having to second guess. Okay, and PMAT stands for prophase, metaphase, anaphase, and telophase. Okay, so in each of these different stages, there's something very specific that's happening in the nucleus of these cells, and we need to know what those different things are. Okay, so let's go ahead and hop into it and see what's happening in the first stage, which is prophase. Okay, remember, prophase is the longest of these 4 different phases, okay? Sometimes we even divide it into an early and a late prophase because it's so long. And there's a lot of different stuff that's going on here. So let's talk about what's happening in prophase. First of all, we're going to duplicate the chromosomes and they become visible, okay? So we've already made that copy of chromosomes, but they're going to condense and they're going to become visible during this state, okay? Also, the nucleolus and the nuclear envelope are going to dissolve. So up until this point, all through interphase, the nucleus is intact, and that nucleus is supposed to keep the DNA safe. But now we're trying to divide the nucleus, and so it has to disappear so that we can get to those chromosomes. Okay, something else that happens is that the centrioles are going to migrate to opposite sides of the cell. So let me just point these out real quick. This is a centriole, these are centrioles. Down here, these are centrioles. They're kind of these barrel-shaped organelles. A lot of the times you'll only see one of them. All through interphase, there's only one centriole in the cell. But remember, in the G2 phase of interphase, we're preparing for mitosis, and so one thing that happens is that we make an extra centriole. What happens is they move to opposite poles or opposite sides of the cells, and you'll see these little lines coming off of them. These lines are called spindle fibers. The spindle fibers are going to attach to the chromosomes, and that's how we can move the chromosomes around. Kind of think of like controlling a puppet with those little strings. The same way you can kind of make a puppet move around by pulling on the strings, that's how we move these chromosomes around, is that they're attached to spindle fibers, and the spindle fibers are attached to the centrioles. Okay, and then that last bullet point there, you can see the spindle fibers span the cell, and that's what these lines are. Okay, so this is the longest phase. Sometimes it's kind of hard to tell if it's in prophase or not. Sometimes you'll see a little bit of a nucleus. Sometimes the nucleus has already disappeared. So this is the longest and maybe most complicated of those different parts of mitosis. Okay, here's a cell. This is a cell under a microscope that is in prophase. What's going on here is you see these really, really dark lines. So those are the chromosomes, and we would know that this cell is in prophase by looking at the nucleus here because we can actually see the chromosomes. Up until this point, all through interphase, you would not see these dark bands inside of the nucleus. Remember, the DNA is in a form called chromatin all through interphase. And that chromatin wouldn't really dye this darkly as it does when it's in chromosomes. So what happens is we take that chromatin, we coil it up, and once we coil it, it's thicker and it's easier to see. So I know that this cell is in prophase because I can actually see some chromosomes here in the nucleus, okay? So that's prophase. Let's move on to the next one. This is metaphase. Metaphase, this one's kind of easier to identify. The main word I want you to remember here is middle. So what's happening in metaphase? Well, the duplicated chromosomes are arranging across the middle of the cell. Sometimes you'll even see people say the equator of the cell, which is kind of that imaginary line that's in the middle. How are we moving these chromosomes around? Well, remember that they're attached to those spindle fibers, and the spindle fibers can kind of move them around. So you can see we've got these 4 chromosomes along the middle of the cell. If you see a cell that looks like this with those dark chromosomes along the middle, you know that it's in metaphase. And middle and metaphase both start with the letter M, so that's just kind of the best way to remember what's going on there. Let's take a look at a real life cell that's in metaphase. Now this one's a little bit harder to tell what's going on than the drawing on the last slide, but to me I can still tell, see these are the chromosomes, and to me it's easy to tell that they're lined up along the middle of the cell. You can kind of see the cell here. This is like the cell membrane. It's kind of like this circular cell. It kind of goes all the way around like this. Well, right here in the middle, these are the chromosomes, okay? And because the chromosomes are lined up in the middle, we know that this is metaphase of the cell cycle, okay? Great work. So let's keep it going. So we've done prophase and metaphase. Let's talk about the next part of mitosis, which is called anaphase. The word I want you to remember for anaphase is either away or apart. And what's happening here is that those chromosomes are going to split and they start moving away from each other, okay? Remember this word right here, the centromeres, okay? Those chromosomes are actually held together. The chromosomes, each side of a chromosome is called a chromatid, a sister chromatid, sometimes you'll see it, but they're held in place by a centromere. And so what happens is when those spindle fibers start to pull those sister chromatids apart, the centromeres split, and then each of those chromatids start to move to opposite sides of the cell, okay? These cells, so like this one and this one, and this one and this one, these are identical chromosomes, okay? The sister chromatids are identical to each other, and they're moving towards opposite sides of the cell because what we're gonna do is split this cell in half, and that makes each new cell having identical chromosomes as what we started with, okay? And you'll also see that the cell starts to elongate. So again, the words that I really want you to remember for this one is away or apart, because the chromosomes start to move away from each other. And actually, somewhat counterintuitively, when they look like, I'm gonna back up just so you can see it. When they look like this whole X structure is a chromosome, okay? It's made up of 2 sister chromatids. But once we start to pull them away like this, now they're each their own chromosome. So this is a chromosome and this is a chromosome. So now each new cell is going to get the exact same DNA as the other one, okay? This is what anaphase might look like in a cell under a microscope. You can see here they started off in the middle, but now those spindle fibers are pulling them away from each other, and so half of the chromosomes are going to go that way and half of the chromosomes are going to come this way. So again, anaphase, the chromosomes are moving apart or away from each other. All right, and then here's the last part of mitosis. It's called telophase. Okay, the word I really want you to remember here is 2, because what you'll start to see here is it's actually easy to see that there are 2 cells. You can see that this is kind of one cell, this is another cell up here. Those 2 new daughter cells are starting to form. So if it looks like 2 cells, you're probably in telophase. Something else to keep in mind here, that telophase is kind of the opposite of prophase. So everything we did in prophase, that first step, it starts to be reversed in telophase. So here are some things that are happening in telophase. First, the spindle fibers dissolve. We don't see those long lines coming off the centrioles anymore. Okay, the chromosomes are going to start to uncoil. So remember, chromosomes are actually chromatin that's very densely coiled up, so that starts to be reversed. Those chromosomes start to relax and moves into that chromatin form. Okay, the nuclear membrane and the envelope and the nucleolus are all going to come back, so a new nuclear membrane forms around the chromosomes. That's kind of what these dotted lines around the chromosomes are. That's the nuclear envelope reforming. And then a new nucleolus forms in each nucleus as well. Okay, so again, telophase, it's kind of the reverse of prophase. A good way to identify it just based off of a picture is just to, if it looks like two cells, is probably telophase. So y'all, great job. Thank y'all so much for following along with me today. Your teacher has some other work. This stuff is complicated, so make sure to give that practice your best effort, and I hope everybody has a great day.